Hello students, I am Dr. Aruna Mohan from Delhi University and I will continue discussing physiology of digestion, the system and the secretions. In the previous session, we discussed about buccal cavity, salivary amylase. Also we talked about stomach, its secretions and digestion in the stomach, and we also studied and discussed the internal structure of stomach. In today's session, we will discuss about associated glands of digestive system, and you already know these glands are pancreas and the liver. Pancreas, which is a mixed gland of exocrine and endocrine function, the exocrine part being pancreatic acini, which secretes pancreatic juices, which are digestive juices. The endocrine part is islet of Langerhans, which will of course secrete hormones like insulin and glucagon. In today's context, we are going to discuss the exocrine part of pancreas, that is enzymes from the pancreas which help us in digestion. Pancreas is present in the loop of duodenum, which is the initial part of small intestine. We know after stomach is the duodenum, and duodenum will continue as small intestine, which is a long coiled tube. The purpose is to increase the surface area. We are going to discuss surface area context little later. Coming to secretions of pancreas again. Pancreas is very delicate organ in our body, and juices are many in number which are coming out from pancreas. Only thing which we all should remember is that some secretions or enzymes are secreted in the inactive form and some are secreted in the active form. For example, trypsinogen is an inactive form, and through the duct of pancreas, when it reaches duodenum and finally the small intestine, by the presence of enterokinase in small intestine, this trypsinogen will be activated to trypsin. So, from pancreas you have trypsinogen, chymotrypsinogen, procarboxypeptidase. We also have amylases, lipases and nucleases. Coming to trypsin again, which is the active form of trypsinogen, it will of course act on proteins. The amylases, you already know about salivary amylase, and now you know about pancreatic amylase. It will act on starch and will convert it into carbohydrate. Lipases, as the name indicates, lipids. Lipases will act on lipids or on fats. So, it is a fat digesting enzyme, but there are more details about the digestion of fat and action of lipases, which we will discuss while discussing liver and the bile. Nucleases, they will act on nucleic acids. So, you can see what a wonderful gland pancreas is. It is giving you all the enzymes for all kinds of food material which we eat. Basically, we are eating proteins, fats and carbohydrates, starches, and we have enzyme for digestion of each from pancreas itself. Suppose pancreas is not there, then you can very well imagine how much of digestion will be affected. So, pancreas being a very important gland present at a very critical location in the alimentary canal and helps in the digestion of food after duodenum. So, students, you have understood about the pancreas, which is present in the loop of duodenum. One more point which I would like to add, that liver and pancreas both open into the loop of duodenum by a duct called hepato-pancreatic duct. Hepato means liver and pancreatic means pancreas. So, liver and pancreas will give its secretions together into the duodenum. We now come to liver. This is largest gland in our body and is very important. It secretes the secretion which is called bile, and please remember, bile is not the enzyme, it has very different role to play. Bile is a secretion definitely, and it is stored in a bag like structure called gallbladder. I am sure all of you have heard about gallbladder, and this bile remains stored there, and when required it comes to duodenum through hepato-pancreatic duct. So, you can very well imagine that in the initial part of small intestine we have pancreatic juice, we have bile, and also we are going to have succus entericus, which is a juice, digestive juice from small intestine. So, in small intestine will have advantage of having bile, pancreatic juices, and small intestine's own juice called succus entericus. Bile is greenish bluish in color. It has bile pigments, bilirubin, biliverdin, bile salts, cholesterol, phospholipids, and of course, it has no enzyme, and it does emulsification of fats. I would like to come into detail of emulsification of fat. I think the main purpose in our alimentary canal as far as bile is concerned is emulsification of fat. What do we mean by this word emulsification? Before I explain this to you, let me also tell you that fat digestive enzyme is lipase with us at this point because pancreas has given it out. Now, lipase will act on fat, but fat molecule is very big and it cannot be dissolved in lipase enzyme, and lipase enzyme cannot enter into the fat molecule. Then how this fat should be digested? To digest this fat, the only way is to break it into small pieces, very fine small pieces, so that each small piece is surrounded by enzyme on the surface of it, and surface part is digested. Suppose it is a very big molecule of fat, then surface area is going to be small compared to if you break it into 100 pieces and then see the surface area, which is going to be very big. So, bile is helping us in breaking of fat molecules into small pieces, small particles, so that lipase can surround it and digest it. That will also mean that lipase will not act unless bile has acted upon, and that is one reason why we are having both the juices together by the same duct, hepato-pancreatic duct, into the duodenum. And from this we also understand that though stomach also had fat digesting enzyme, that is stomach lipase, will not act upon fat till the food reaches to the small intestine, till the bile is made available. That means the fat digestive enzyme secreted by stomach will also act on food, the fat part of the food, only after it reaches the small intestine. You can understand that how important bile is. Suppose somebody does not have bile, or gallbladder is removed, or a liver is not functioning properly, then that person cannot digest fats. Sometimes there are stones in the gallbladder, so doctor will remove whole of the gallbladder along with the stones. But in this case, liver is functioning, some bile is secreted and some portion of it will reach duodenum. So, little help in emulsification of fat will be there. But if liver is damaged or acted upon by some alcohol or some other drug, and hence it is not functioning at all or not producing bile at all, there is no way that we can digest fats. And you can very well imagine when we eat fat and it goes as such till the rectum, and how difficult it is going to be for the alimentary canal. So, the main role of bile is emulsification of fat, so that lipase can act upon these small fat molecules and digest it. When I talked about pigments, biliverdin, bilirubin, bile salts, let me tell you, the color of feces, which is brownish, bluish, greenish in color, is because of these bile salts and bile pigments. This shows you the way liver and pancreas are present in our abdominal cavity. You can see the huge liver lobes. You can also see greenish color gallbladder, and you can see delicate pancreas in the loop of duodenum, and then you can see a common duct, hepatopancreatic duct, which is making its way to the duodenum, and the juice from both the glands are delivered to the duodenum lumen or lumen of the duodenum, mixed with food, and goes down to small intestine along with the secretions. At this point, I should also tell you that there is a pace with which food moves in our alimentary canal. We of course call it peristalsis. When you eat food, that remains in the stomach for 4 to 5 hours, minimum 3 hours, and that is why it is advised that do not eat very frequently. Suppose you have taken lunch at 1 o'clock. Now it is going to be there for minimum 3 hours. Digestion begins in stomach. In 1 hour, digestion has taken place, little bit, not full, but then you eat something again after 1 hour. That means what you have eaten after 1 hour, that is in the first stage of digestion, and what you took 1 hour before is in the second stage of digestion. Now after half an hour, you have eaten something else again. So again there are 3 stages, first, second, third. So we are confusing our stomach. So it is advisable that have a good meal and then don't take anything solid at least for 3 hours. You can have some liquid, some juice, some milk, tea, or at the most some fruit, but don't take main meals without the gap of 3 to 4 hours, because that food is remaining in your stomach. Then it will move to duodenum with a speed, particular speed, which is the speed of peristalsis, and then it will move to small intestine, where it will remain for a long time, because digestion is continuing. After bile, after pancreatic juices, digestion continues, and small intestine will have its own juice, succus entericus, digestion continues, and after that absorption will also take place, and hence food will remain for very long time in the small intestine. We have discussed emulsification of fats. I would like to give you a few more points regarding emulsification. We know emulsification means breaking down of fats into small micelles. This is one thing. Second thing, bile activates lipases. I would like to enhance this point regarding bile action. I did tell you that bile does emulsification of fats, breaking it into small pieces. I also told you lipases can act only after emulsification has taken place. Now I am adding one more point to this, that bile has 2 major roles. One is to emulsify the fats, other is to activate the fat digesting enzyme lipase. I take you back to the secretion of lipase by stomach, by pancreas, and of course by small intestine also. But this lipase will not act on fat till bile has emulsified it. One point. Second point, after emulsification has taken place, this bile will now activate lipase, and now this lipase, which is in the active form, will act on fat molecules which are in the form of micelles now and will digest the fat. So, bile is helping fat digestion in two ways. First, emulsification. After emulsification, activating lipase, the fat digesting enzyme, allow it to act on fat molecules and digest it. So, bile takes total care of fat digestion in our alimentary canal. With this, we come to the end of this session. Thank you.